Genomics has moved from a specialist research discipline to a central technology driving medicine, biology, and biotechnology. Over the past decade, advances in sequencing chemistry and instrumentation, single-cell and spatial methods, genome editing, and computational approaches have expanded what can be measured, manipulated, and interpreted, and have begun to reshape clinical care, drug discovery, and agriculture. Sequencing and assembly short-read sequencing became ubiquitous for population-scale studies, but the biggest technical leap has been the maturation of long-read and highly accurate long-read platforms. Pacific Biosciences' HiFi reads and Oxford Nanopore continuous improvements enable contiguous, haplotype-resolved assemblies that reveal structural variants, repeat sequences, and complex loci that short reads miss. These advances, together with pan-genome efforts that move beyond a single linear reference, are producing more representative and complete references for humans and other species, improving variant discovery and reducing biases in downstream analyses. Single-cell and spatial genomics. Single-cell RNA-seq, ATAC-seq, and multiome assays now profile gene expression, chromatin accessibility, and other modalities at cellular resolution across thousands to millions of cells. Concurrently, spatial transcriptomics and in situ hybridization technologies map gene expression directly in tissue context, preserving architecture and cell-cell relationships. Combining single-cell and spatial data with protein-level and epigenetic readouts, spatial multiomics, is enabling mechanistic insights into development, immune response, tumor microenvironments, and neurobiology. Epigenomics and direct modification detection sequencing platforms increasingly capture epigenetic information directly. Nanopore sequencing profiles DNA methylation and other base modifications during readout, while improved bisulfite-free methods expand methylation mapping. Single-cell epigenomics and cut and tag/cut and run methods provide high-resolution views of chromatin states, histone marks, and transcription factor binding in heterogeneous samples. Genome editing and therapeutics. CRISPR-based editing tools have diversified and matured. Beyond classic double-strand break approaches, base editors and prime editors enable precise nucleotide changes with reduced reliance on cellular repair pathways. Delivery technologies, including lipid nanoparticles, viral vectors, and improved ex vivo workflows, have advanced translational pipelines. Clinical applications have progressed rapidly. Ex vivo edited cell therapies for hematologic disorders and in vivo editing for genetic diseases have shown promising results, demonstrating both efficacy and tractable safety profiles in early studies. Gene-modulating modalities, RNA therapies, antisense oligonucleotides, and engineered cell therapies broaden the therapeutic toolbox. Diagnostics and rapid response. Genomic sequencing-based diagnostics are accelerating. Metagenomic sequencing for pathogen detection, rapid viral genome sequencing for outbreak surveillance, liquid biopsies for cancer detection, and cell-free DNA assays for prenatal and transplant monitoring. CRISPR-based diagnostics, e.g., collateral cleavage assays, offer low-cost, point-of-care nucleic acid detection options that complement sequencing in resource-limited settings. Computational genomics and AI. The scale and complexity of genomic data have driven major advances in computational methods. Deep learning and generative models now predict functional effects of variants, regulatory element activity, splicing outcomes, and, crucially, protein structures, the latter transformed by models such as AlphaFold and related systems, which accelerate functional interpretation and drug discovery. Integration frameworks for single-cell and multi-omic data, scalable cloud-native pipelines, and improved variant annotation tools are essential to translate raw data into actionable insights. Applications across sectors in medicine. Genomics informs precision oncology, risk stratification for inherited diseases, pharmacogenomics, and microbiome-based interventions. In agriculture, genome-enabled breeding and gene editing accelerate trait improvement and resilience. Conservation genomics uses whole genome data for population management and biodiversity monitoring. Synthetic biology leverages genomic design to build novel pathways and organisms for materials, fuels, and therapeutics. Challenges and ethical considerations. Rapid technological progress also raises practical and ethical challenges. Data volume, standardization, storage, and compute costs strain infrastructure and require interoperable standards and secure cloud frameworks. Ensuring equitable access to genomic medicine, protecting genomic privacy, and addressing potential misuse of gene editing technologies are urgent societal issues. Regulatory frameworks must balance innovation with safety, especially for germline interventions and ecological release of edited organisms. Future directions expect continued convergence of technologies, more routine clinical-grade long-read and direct RNA sequencing, integrated single-cell spatial multiomics at scale, improved in vivo delivery systems for safe, precise editing, and tighter integration of AI to predict phenotype from genotype. As references diversify and datasets grow, personalized, mechanism-based interventions will become more practical, while governance and equitable deployment will determine how broadly society benefits. Genomics is no longer only about reading DNA, it is about understanding function, context, and causality, and about responsibly using that knowledge to improve health, food security, and our understanding of life.